Okay, so in this ENIGMA-U module regarding ethical issues in behavioral science, I'll talk about the replication crisis and issues of confidence in science. And this is a relatively new issue, probably over really taking a firm hold of the sciences over the last decade or so. So, what is the replication crisis? It's this relatively recent movement in science that's brought to question some traditional scientific practices. And it's been triggered by recent work showing that some long-standing classic findings could not be reproduced in a number of scientific disciplines. And it started in particular in the area of the social sciences. And the motivations for the crisis are not new. It dates back over 60 years or longer with Paul Meehl, a clinical psychologist, and Cohen, a statistician who had concerns about our emphasis of null hypothesis significant testing. This is not a new issue, but it's something that's now taking hold in a very new way. So it's openly discussed, and there's actually research about it, including the growth of an area of meta science in data sciences. So, what is a replication, and why is science in crisis in general? Replication is testing a prior finding with different data. And so you have a finding, you have your own data, you want to see if you get the same finding. You'll hear also terms like robustness, which is testing the reliability of a prior finding using the same data, but a different analytical strategy. So it's showing that the effect, the ultimate outcome is quote unquote robust to how we analyze it. So you have same data, different analysis, and then reproducibility is testing the reliability of a prior finding, use the same data and same analysis, but with a different investigator. And so these are all highly related to one another, and sometimes they're used interchangeably. But really, ultimately, what we're talking about is the reliability of a finding. How much faith can we have in a finding, whether it be the same data, different method, or different investigative team? In the behavioral sciences, a paper in 2015 really sparked a lot of discussion, in particular in the area of psychology and social psychology, where investigators set out to test 100 really well-known studies in the area of social psychology that were sort of anchors or important studies for the field. And so they chose these 100 studies and they set out to test them to see how reliable those studies would be. And ultimately, unfortunately, they're only able to replicate 39% of those studies. So only 39% showed similar results to the original finding, which is much lower than anybody had anticipated. And replication results left 68% with statistically significant effects. So the original study, then this is a graph showing the effect size of the original study, which is in light green. And then pink is in the replication size effect. And you can see that the power or the effect is much lower in most of the studies during the replication. The mean effect size of the replication was about half the magnitude. And 97% of the original studies reported significant results. 36% of the replication had significant results. What does this speak to? The 97% in particular is it's really hard to publish a paper without significant results, which is something we're reinforced for as scientists. Go find the significant result. That within itself is a problem. And it has been referred to as quote unquote P hacking, finding P is less than 0.05, where the result is meaningful or significant. And those times when the result is not significant, often we can't publish it. So we just move on to the next study and leave that study alone, which is unfortunate because it's still a study that's been conducted and it could have been a well-designed study that didn't find an effect. That within itself should be known. So the sources of the replication problems are a devotion to null hypothesis significant testing, like I just described. I've got to find a result. I've got to find something significant here anywhere, or I can't publish my paper. Small sample sizes. This is really important, in particular in the clinical neurosciences, where we use complicated methods like imaging and genetics that can be invasive or they can be hard to use. And so sample sizes may be quite small. The number of participants may be small. And that makes it really hard to see some effects. Flexible hypotheses or post hoc explanations. And this is not to indicate that scientists are being dishonest. It's that over the course of a study, our expectations about the data and the findings mature, they change. We get a better idea about what's going on. And so our original ideas may not match what we see later. And we may, in essence, sort of mold our ideas and our hypotheses to the ultimate outcome. So the ultimate outcome looks like we predicted it all along, when actually, if we had gone back to the original, the beginning of the study, we may not have expected that. And so what that means is it may have just been a chance that we found something, but we explain it away as part of our hypotheses. Failure to falsify, which is related to this. And that means that we didn't set up our hypotheses to begin with in a way that could truly be tested, so that if A is the outcome, then the hypothesis is clearly tested. And if B is the outcome, then the hypothesis is clearly refuted. A hypothesis, unfortunately, can be somewhat murky. They can be supported in a number of different ways, and that makes them difficult to falsify. And then there's lack of replication efforts, straightforward. And that meaning very few studies set out to replicate prior results. And the reason for this is that it's challenging. You're not rewarded for this as a scientist historically, although there's efforts to change that. That replicating somebody else's findings is not innovative, it's not creative, it's not advancing science, it's simply a replication. And so the scientific reward, professional reward, has been minimized for replication efforts for a long window of time. And there's now groundswell efforts to change that. So what is the crisis itself? It's not just in the social sciences like I just described, it's across virtually every discipline of science. It's in cancer biology, it's in economics, it's definitely in biology, it's in psychology. And so it is, because we're all human in all of these disciplines, we are imperfect. And so and the scientific endeavor has unfortunately been rewarded for a subset of the behaviors like publishing and granting and novelty and innovation over getting it right and making sure that the finding is you can hold it up multiple ways and you can interrogate it using a number of methods and it still stands there. And so I identify and replication is part of that, which has not been rewarded typically. And so in a really nice paper in Nature, they surveyed scientists and they asked 1,500 researchers, is there a reproducibility crisis? And 38% said a slight crisis, 3% said no, 7% said don't know, 52%, half, said there's a significant problem here. And that's important to pay attention to. The scientific community has recognized this. And at least if you look at a slight crisis, although defining a slight crisis is a little challenging, but at 90% of scientists will say we need to do something about this. And that's the encouraging part. And when asked the question, have you failed to reproduce an experiment, either someone else's or my own? Again, this gets back to the point I made a moment ago. It's not discipline specific. All areas, earth and environment, medicine, physics, biology, chemistry, areas that you think would have a very high replication rate, we find can be surprisingly low. And so the replication crisis does not discriminate across the discipline. 70% of the researchers have tried and failed to reproduce another scientist's experiment, and more than half have failed to reproduce their own experiments. So how did we get here? And so one is the incentives for individual success are focused on getting it published, not necessarily getting it right, which I spoke to a minute ago. And so there's an entire ecosystem within science, the universities, publishing funders, and societal need. And scientists just play a role sitting in the middle of that. And so This really good paper by Manofo in Nature Human Behavior looks at this sort of cycle of life, and they're sort of feeding into bad practices, feeding into one another. And so the first is lack of replication. There's also lack of data sharing, flexibility and analysis. So a lot of different ways we can analyze our data, which lead to a lot of different outcomes. Exploratory research, which may be presented as confirmatory, that we thought that all along and now we've confirmed it, as opposed to this is something we might look at, but it needs confirmation. And the file drawer problem. Let's start with that one. So the file drawer problem is something I spoke to a minute ago, and that is if there isn't a significant finding in most studies historically, you couldn't publish it. There would be really no journal that wanted a study that would be referred to as quote unquote failed. When in essence, science is about discovery. It's not about discovery of that there is a quote unquote statistically significant finding. It's about understanding a phenomenon and understanding the relationship between variables. Even if that means something you expected to see was not there, and that doesn't mean that's not something interesting. Now, sometimes we don't see effects because we're underpowered, the sample size is small, the design is not so fantastic, and those are all reasons why a paper shouldn't be published. But it should not be strictly because p is there was no significant finding. P is greater than 0.05. And so the file drawer problem is the cartoon here says, well, this is where we put the non-significant results. And in the fate of 224 social science experiments, most null results are never written up, which leaves us in this really awkward space of individuals perhaps conducting similar studies that are never written up because the findings are null, where science would be much more efficient if those findings were published. Flexibility in analyses is another that's really important in our own area in imaging. There are 60,000 ways, literally 60,000 ways to process our data, which is far too many. It makes it really challenging to know at the end, if any of those decisions influence the data. There's significant effort right now to clean that up. There's also the sort of flexibility in data collection analysis considered has been called the garden of forking paths, meaning all of the little decisions you make as an investigator add up over time in order to get you to that p is less than 0.05. But if you look at the tree on the right there, maybe one of those is p is less than 0.05, meaning a significant result, something that would be published, but many of the other trees would not have been. And so it sort of depends on what pathway you took. And so there are, again, efforts for us to understand not just the one pathway, but let's look at all of them simultaneously, and advanced computational methods have allowed for that. And so our analyses decisions that go unreported, did we exclude outliers? Is it median or mean? Do we control for time? How do you define some of the variables? And how many of these paths did you try but didn't report because you didn't get p is less than 0.05, which is really important. So there's too many degrees, quote unquote, degrees of freedom in the analysis pipeline. Exploratory research presented as confirmatory. I talked a little bit about that. So hypothesizing after the results are known is called harking. And so you just at the outset, there's two different contexts. On the left, there's a context of justification. Here's why we want to do it versus a context of discovery. And then there's the step of confirmation on the left versus exploration. And there might be data independent. You're doing that with independent data versus on the right, there's data dependencies. They actually they rely on one another for a finding to exist. And then on the left, it'd be hypothesis testing, and on the right is hypothesis generating. On the left, because of it's more definitive in its nature, you could say that the p-values, they may hold stronger value, they may be interpretable. On the right, it's really more about discovery, and it's less about whether or not we have a statistically significant result that can drive science. It's more about understanding the phenomena at the basic level so that we can begin developing hypotheses. But The problem is often the approach on the right is posed as though it was on the left. So instead of discovery and exploration, it's written and published as a confirmation. And so presenting exploratory as confirmatory increases the publishability of results at the cost of credibility of the results. And so that brings us to pre-registration. Pre-registration is, I think, the most useful tool that's been developed, or that's perhaps not developed, but is gained increased visibility and use in order to combat issues around the replication crisis. And the idea is the following. You, at the outset, you write up your study, the method, before you look at any of your data. You propose the analyses to test the hypotheses again before you look at your data. You make that publicly available so everybody can see it. And so the study plan, hypotheses, data collection, the measured variables, statistical models, inference criteria, and then you post that for the Center of Open Science or other avenue for that support pre-registrations. This helps a lot with regard to breaking that cycle of the scientific process that leads to replication problems. First, it helps with the file drawer problem, because if you don't have significant results, but it was a really good study, it was powered enough, and it was evident on the front end, you tested exactly the way you said you were going to test it, the phenomenon, then it should be published. And I think reviewers are increasingly open to that. That it's not that you ran 300 different analyses and there was nothing there. It's that you ran exactly what you said you were going to do with a well-designed study, and there was no finding. That is part of the scientific endeavor that should be published. We need to know about it. P-hacking, or unreported flexibility in data analyses, it helps definitely with that. And it also helps with hypothesizing after the results are known, because you didn't see the data and your hypotheses are publicly known. Back to the research life cycle, lack of replication. This goes back to one of the comments I made earlier, that efforts for conducting replication science have not been rewarded as much in the scientific community, or even at the university level, or research institutes, and that is changing. And lack of data sharing. Data sharing does a number of things, including increasing our sample size, gives us greater power, increases our expertise, and the way in which we can conduct our science. So in a quick kindergartner's guide to improving research, there's really only a couple of things that we can do. There are many things we can do, but two quite simple things we can do that can help a lot. So pre-registration is part of showing your work, and the other is sharing. And so pre-register your studies and your analyses plans, and the Center for Open Science supports these. And also, to the degree that you can, keep your, as far as pre-registration, but also your analytical pipelines and your code, make them as open source as you can. And that allows other investigators to help, to comment, to contribute. And that really, the team science approach changes the tenor of science away from, this is my data, my career, my concern, to we are all, in some ways, we all have the same goal. And a collaborative effort can get us there faster. So I talked for a moment about the challenges with publishing only findings that are statistically significant, and also the lack of reward structure for being for data sharing and being transparent and open with your code and your data pipelines. And there are current efforts, for example, the Open Science Award. It's awarded annually by the Wellcome Trust in England, NIH, and Howard Hughes Medical Institute to quote unquote unleash the power of open content and data. And this is an encouraging development. It's an indication that the scientific community is taking open science and data sharing seriously. And it's trying to change that reward structure away from Um, Publish more papers. This is your lab, your data, your space, and team science can't get you there. It's trying to change the reward structure so that um, team science, greater collaboration, greater sharing is more rewarding, both for the scientific endeavor, but also for individual labs and careers. So the pre-registration, open materials, open data, and open access. Even when we publish our papers, we try to make them open access. So anybody on a computer with Wi-Fi anywhere in the world can click on it and gain access to it. So in summary, how can we address the replication crisis? Pre-registered studies is one of the best steps right now. Also, though, what goes with that in your pre-registration is attempts to falsify existing hypotheses, publish your null findings, and if you have a pre-registration, it really helps with that. And definitely data sharing. You will open up collaborations you never thought were possible through data sharing. If anybody ever reaches out and says, hey, I have an idea, would you mind sharing your data? Definitely meet and listen. Now, not all collaborations work out, but it's definitely worth listening. Transparency in methods is really important. To all the steps we took, even those where things didn't work out, that's okay. Adoption of a replication culture. It's important that our studies set out to replicate really critical findings that are foundational to our understanding of the phenomenon. We have to see it again. And we as scientists need to demand that. We need to demand to be rewarded for that, and it must be part of our science. And an advocate for culture of openness and transparency in science. The idea of team science is taking hold in some corners of in different scientific disciplines, and that's encouraging. Hope it continues. So summary, replication crisis is real and alive, a legitimate issue being addressed across scientific disciplines. Emerging with this crisis, growing recognition of the ways that current scientific approaches can be improved. The failure to replicate results in science does not indicate that science is broken or should not be trusted. It's a human endeavor and therefore imperfect. The alternative is not no science, it's better science. And there are a lot of ways in which the scientific community has taken the replication crisis very seriously and working to create better science.